Hi, Dr. Biology here, and this is a video on DNA and the genome. So this is related to inheritance, variation and evolution, which is part of GCSE biology and combined science. So this is part 2 of a series of videos on this topic. So firstly, if we look at a cell, if we look within a cell and the nucleus, we can see that there are chromosomes, and chromosomes are wound up pieces of DNA, and a section of DNA codes for a gene. So what is DNA? Well, DNA is the genetic material in the nucleus of a cell, and DNA is a polymer, so it's made up of 2 strands that form a double helix. So the DNA is contained in structures called chromosomes. DNA determines what inherited characteristics that you have. Chromosomes, well, DNA is found in chromosomes, and chromosomes, they come in pairs. So in a human there are 23 pairs, one pair of the sex chromosomes. So for example, in a male XY and in a female XX. So what is a gene and how are proteins made? Well, a small section of DNA codes for a particular sequence of amino acids to make a specific protein. So a piece of DNA, part of the DNA code of a gene, will code for an amino acid that makes a specific protein. So genes simply tell the cell what order to put the amino acids in. So this diagram shows the gene and the section of DNA containing those instructions that make amino acids, and those amino acids are joined together by bonds and therefore they make a protein. So a genome is the entire set of the genetic material in an organism. Now the human genome was actually first sequenced and completed in 2003. So scientists can identify genes linked to disease and develop effective treatments. You can also trace the migration of populations of people around the world, as all modern humans are descended from a common ancestor who lived in Africa. So DNA structure. So this is GCSE biology only. So a piece of DNA is known as a nucleotide. Now, DNA is a polymer and it's made from 4 different types of nucleotides. So each nucleotide consists of a common sugar, phosphate group, and one of 4 different nitrogenous bases attached to the sugar, and those are known as A, C, G, and T. Now 3 bases, so 3 of those bases will code for 1 amino acid, and that's known as a triplet code. DNA has the bases are what we call complementary base paired. So what we mean by that, A always pairs up with T and C always pairs up with G, as you can see in this diagram. So you can see the double-stranded piece of DNA at the bottom right hand corner, and you can see the A to T, C to G. There we go. So A always pairs up with T, C always pairs up with G. There are also, in the DNA code, there are non-coding parts. Now, you will need to know this for GCSE biology higher tier only. So there are parts of the DNA that don't code for a protein. So those are called introns. So there are large parts of the code that don't actually code for proteins. Scientists believe that these switch genes on or off, so they can control whether a gene is expressed or a protein is made. And currently there is a lot of research into this part of the DNA code, known as non-coding DNA or intron. Protein synthesis. This is in GCSE biology higher tier. So how on earth are proteins made? Well, first of all, in the nucleus the DNA code is copied, and that produces a molecule called mRNA, or messenger RNA. That then travels through the nucleus and into the cytoplasm and eventually to the ribosome. So this is where proteins are synthesized. So the mRNA codes for a particular amino acid, and that's usually 3 bases, so 3 bases of mRNA, and they code for a specific amino acid. They're put together and they're bonded and they make a protein, and that folds up to make a unique shape, and it forms proteins that do their jobs, such as things like enzymes, hormones, or forming structures in the body such as collagen. Now, mutations also occur. So a mutation is a random change of DNA. So some mutations can be inherited, others occur randomly. Sometimes substances or types of radiation can increase the rate of mutations. So mutations change the base sequence of DNA bases, so the amino acids may be different, meaning that sometimes the protein it codes for is what we call non-functional. So in the diagram you can see an enzyme here, and you can see that a mutation has caused the enzyme active site to change shape. If it changes shape, it no longer fits the substrate, therefore you no longer have enzyme substrate complexes. There are different types of mutation, and again you will need to know this for GCSE biology higher tier. So the first one is substitutions, where one base is swapped for another base. So you can see the substituted bases, and what sometimes happens is that the amino acid it coded for is made to be different. Sometimes it's the same, but sometimes it's different, and therefore affects the functioning of the protein. Second is deletion, so a random base is deleted from the sequence. This is more this causes more issues because it shifts what we call the reading frame from the left of the one base. So it changes all of the codons or all the triplet codes after it, and therefore all the amino acids may change, therefore making the protein non-functional. And thirdly, insertions, where a random base is added to the sequence. So similar to deletions, the reading frame has a knock-on effect further down the sequence, and therefore has a serious effect on the protein. Okay, so let's have a look at some questions. So what I would like you to do is to pause the video, read and answer the questions, and then come back and I will go through the answers with you. So pause now. Okay, so this question is asking several questions, so here we go. A gene is a small section of DNA. The sex chromosomes in the human male are XY, X and Y. Most human body cells contain 46 chromosomes, and the number of chromosomes in a human gamete is actually half the number in body cells. Gametes are produced by meiosis. Okay, this question is a bit more difficult, so again, what I would like you to do is to pause, read and answer, and then come back and play the video. Okay, so A, in which part of an animal cell is DNA found? That is the nucleus. The letters A, C, G and T, they are known as bases. One strand of the DNA in the section labelled X contains the following sequence of these compounds. How many amino acids would this section of the DNA code for? Remember, they're in triplet code, so 3 bases, so it would be 4. So there's 12 bases, so 3 times 4. And then last one. Okay, so they code for a sequence of amino acids, or the order of 3 bases, which codes for a protein. So you don't need all of that answer, you just need, if you look at the /, that means or. Next question. Okay, this is biology higher tier only. So again, I'd like you to pause, read and answer. Okay, so this is a bit more difficult, this question, so let's look at the answers. So basically, word used to describe a small section of DNA is the gene or allele. In the cell where a protein is synthesized, that's the ribosome. And then describe how the protein for blue-eye color is synthesized. You can have any 3 of the following. So amino acids make up proteins, or the sequence of amino acids, or bases form a code, or you could say bases work in threes in a triplet code, and they code for one amino acid. D is about mutations, so it asks what would happen to the structure of the protein synthesized by this part of the DNA molecule. So it's asking about, it's looking at the W and X base, so the different wrong amino acid would be coded for. So what might the effect have? Well, this code was for eye color, so you would get a different eye color, or you could say, give an example of a different eye color. Okay. I hope you found that useful. Please do subscribe to Dr. Biology, and there are more videos on this section of work coming soon. Thanks very much, and I'll see you soon.